This portion of chapter 15 focuses on the ear. So we're going to talk about the structures of the ear. We'll also talk about how you hear, along with equilibrium and balance, because remember, you do have some structures in your ear that are responsible for that. So before we really get into the different structures of the ear, let's talk a little bit about how we divide the ear up into three major areas. So one area is the external ear. This is only involved in hearing. We also have the middle ear, and the middle ear is also only involved in hearing. And then deep in the ear, embedded within the bone, is the internal ear. This one is involved in hearing, but this is also where we find those structures involved in equilibrium or balance. So let's walk through these three different areas of the ear. I'll show you where they are on the ear. So the external ear, you can see here, is basically everything that you can touch or put a Q-tip in. Okay, so that's the external ear. The middle ear is everything from the eardrum, which we call the tympanic membrane. And behind the tympanic membrane, there are these three little bones that we call ossicles. And there's also a tube here that actually drains down into the back of your throat. We used to call that the eustachian tube, but now we call it the pharyngotympanic tube. And then lastly is the internal ear. And the internal ear is the deepest portion. It's actually embedded within the bone. And it contains structures like this one right here. It's called the cochlea. It looks like a little snail shell. This one is involved in hearing. And it also includes the structures over here called the semicircular canals. And those are involved in balance or equilibrium. And so that's why we say that the internal ear is involved in not only hearing, but also balance. So let's go through each of those major areas of the ear and talk a little bit about the structures we find there. So the external ear, again, this is everything you can touch or put a Q-tip in. So the pinna is the funnel. This is on the outside. It's what you would put earrings on. This is a little funnel that helps to actually funnel the sound waves into your auditory canal. And the auditory canal, if you remember from our bone markings, we call this the external acoustic meatus. Sometimes we also call it the external auditory meatus. Same thing. This is basically a tube that's aligned with sebaceous glands and ceruminous glands. Now remember that term cerumen. We looked at this way back in the beginning of the semester. Cerumen is basically earwax. So these are the glands that secrete earwax. And the whole focus of the pinna and the external acoustic meatus is to help transmit sound waves so that they can hit the eardrum. So the external ear ends at the eardrum, which we call the tympanic membrane. This is really the boundary between the external and middle ear. The tympanic membrane is incredibly thin. It's made of connective tissue. And when sound waves come into your ear, they hit that eardrum and it causes that eardrum to vibrate in response to those waves. Now we know that sound travels in waves. If you have ever been next to a car that has their bass turned up really loud, you can kind of feel it. You feel it in your car vibrates. Sometimes like your rear view mirror might vibrate, and that is the sound waves that are traveling. From there, your sound waves will move from the eardrum through those little tiny bones that are right behind the eardrum called the ossicles. So let's look at an image again. So those sound waves are going to come in and they're going to hit that tympanic membrane. Think of a drum, right? If you were to hit a drum with a drumstick, it's going to vibrate. So it's the same concept. That eardrum, that tympanic membrane will vibrate. And behind the eardrum, remember, we have three little tiny bones. These are called the malleus. This is the first one. It touches the tympanic membrane. The incus is the second one in the series. And the third one, it looks kind of like a horseshoe, is called the stapes. And those sound waves are going to actually cause those bones to vibrate, okay, from the malleus, the incus, and the stapes. And again, those little bones are called ossicles. Now, also within the middle ear, we find something called the pharyngeal-tympanic tube. This used to be called the eustachian tube. Now it has this really long, complicated name for a reason. This name actually describes where it starts and where it goes. So this is a tube that runs from that tympanic membrane, that eardrum, all the way down into the pharynx. So that's why it's called the pharyngeal-tympanic tube. So it's basically connecting your middle ear down into the nasopharynx. This helps to equalize pressure. You know, when you fly on an airplane, they'll tell you to chew gum or yawn, open your mouth. You're opening your mouth to try to equalize that pressure, to get that air to equalize in your ear. This also is a tube where when someone gets an ear infection, a lot of times ear infections, especially for little kids, they actually start as sore throats. And from there, the bacteria can move from that pharynx all the way up the pharyngeal-tympanic tube into the ear, into that middle ear. And we'll look at some pictures of that in just a little bit. The pharyngeal-tympanic tube, you can see it's right here. They don't show you that tube draining all the way down in the pharynx, but it does. Now let's talk a little bit about ear infections. We call this otitis media. This actually stands for a middle ear inflammation. Very common in children, again, because they get a lot of sore throats. The bacteria very easily can move up that pharyngeal-tympanic tube. The pharyngeal-tympanic tube is also a little more horizontal in children. Their head hasn't elongated and the tube has not become more vertical yet. And so the bacteria and the pus just kind of hang out in that pharyngeal-tympanic tube. Now, typically ear infections, they're really painful because what happens is pus builds up behind that eardrum. So let's go to this picture. Pus will build up back here. So those bacteria move up from the throat to that middle ear. That pus builds up and it puts a tremendous amount of pressure on that eardrum, and it causes the eardrum to start bulging outward, and it becomes really, really painful. With otitis media, most often it's treated with antibiotics. They'll look in with an otoscope and they can see that the eardrum looks white and opaque. They can see all the pus behind it. And so a quick, easy treatment are antibiotics. The problem is a lot of children have constant reoccurring ear infections. And this is a really frequent cause of hearing loss in children. And the reason, I'll go back to this picture, is because if you constantly have pus back here in this middle ear, it actually causes these little ossicles that we talked about here to start fusing together. And if they fuse together, then they don't vibrate very well, and it causes a reduction in hearing. So that is hearing loss. Now, it used to be before antibiotics, how they would treat an ear infection is through a laryngotomy. They would basically take this really long, sharp, pointy probe, stick it in through the outside of the ear, and they basically would rupture the eardrum. The beauty of that is that it would relieve the pressure. It hurts initially, but then the pressure's relieved. The pus can drain out of the ear. The problem is with repeat lancing of the eardrum, can cause the eardrum to get thicker, can build up scar tissue. That can also decrease hearing. So instead, now what we do is ear, nose, and throat doctors, if children are having constant repeat ear infections, they'll put in tubes. So if you've ever heard of a child having tubes put in their ear, you are looking through the ear canal, and you can see this clear film in the background. That is the eardrum, and you can see that bright tube right there. This is basically doing the same thing as a laryngotomy. They lance the eardrum, they insert this little plastic tube. That plastic tube will eventually fall out, but in the meantime, it's holding that eardrum open so that as pus builds up behind it, it can quickly drain. And this is going to help to prevent those ossicles from fusing together. Now I do want to talk more about those little ossicles. Again, there's 3 of them. There's the malleus, the incus, and the stapes. And again, when sound waves come in and hit the eardrum, it causes a vibration in those ossicles. They're going to vibrate as well. Now let's talk a little bit about what we find in the inner ear. The internal ear is the structures of the internal ear are embedded in the bone. They're embedded in the temporal bone. And so we find 3 regions within the internal ear. There is the vestibule, the semicircular canals, and the cochlea. The cochlea is the structure here. It looks sort of like a snail shell. This is involved in hearing. The semicircular canals are here. They are filled with fluid. They're involved in balance. And the vestibule is just the area between the two. So I always think of the word vestibule like waiting area. So this is just a little waiting area between the semicircular canals and the cochlea. These are filled with what's called paralymph. This is very similar to cerebrospinal fluid. It is made from blood filtrate. Now I want to talk about each of those 3 structures. We're going to start with that vestibule. The vestibule has 2 little sacs within it. There is an area called the saccule, and then there's an area, this is the saccule, and then there's an area called the utricle. The saccule is, you can see closer to the cochlea over here, so it's continuous with the cochlear duct. The utricle is continuous with these semicircular canals. And these sacs are going to house equilibrium receptors. They help to respond to changes in the position of the head. So rotational movements, for example. Now the semicircular canals, which look like semicircles, these are filled with fluid. They actually lie within 3 planes of space, and they have receptors that respond to angular and rotational movements of the head. And again, these are continuous with the utricle, so they're responding and communicating to the utricle about movement. Now the cochlea is this snail shell looking structure. It's a spiral bony chamber, and this image makes it look really big. It is not. It is very tiny. It's the size of about a split pea. And again, it's continuous with this little structure over here called the saccule. The cavities within the cochlea are filled with that fluid called paralymph. And inside the cochlea, there is this flexible membrane. It's called a basilar membrane that plays a huge role in sound reception. So it picks up sounds and it sends the sound back through the cochlear nerve. And the cochlear nerve is going to take that sound information all the way back to the brain for processing. Now, let me go back to this. We didn't talk about a cochlear nerve as a cranial nerve, but we did talk about the vestibulocochlear nerve. And so you can actually see here, you can see that portion coming from the vestibule, and then you can see that portion of the nerve coming from the cochlea, and you can see where they merge together. So that is the vestibulocochlear nerve. This is a great picture showing you what the inside of the cochlea looks like. So what I want you to look at here is you can see all of this in purple. This is all paralymph. So it's openings filled with paralymph. And then if we look at this image here, we can see this little box, and let's blow that up really big. So that's what we're looking at here. Something nice and big, and you can see this flexible membrane called the tectoral membrane. So what happens is sound waves are going to come into the ear through that ear canal. They're going to hit the tympanic membrane. The tympanic membrane will vibrate. It sends those vibrations through the 3 ossicles. Then those vibrations move back into the cochlea. When the vibrations hit the cochlea, because all of this is filled with fluid, the tectoral membrane will start to move and vibrate as well. And you can see what sits underneath. Here's the membrane. What sits underneath the membrane are these little tiny hair cells. So you can see here's a cell, here's one, and here's one. They have little hairs on top. So you can see those little cilia. And every time that tectoral membrane hits against those cilia, it triggers an impulse going back through that cochlear nerve. And so that's what this slide explains. It talks about those vibrations, again, moving all the way through the ear and hitting the eardrum, transmitting through those ossicles and into the cochlea. That tectoral membrane will start to move, and it will hit those little hair cells and send that signal back to the brain. And this is also a great example of that. So it is showing you those sound waves coming in, hitting off of the tympanic membrane, moving through those 3 ossicles and all the way back into the cochlea. I like this image as well because this one is showing you, I like the little yellow here. It's actually showing you that tectoral membrane, and it's showing you when those hair cells get stimulated, and that is going to send that signal back to the brain. So how do you distinguish different pitches? How do you distinguish loudness? How we distinguish pitch is pressure waves in the cochlea are going to cause that membrane to vibrate. This is going to stimulate those hair cells. But the membrane actually ranges in width and stiffness. And so what we see is that narrow, stiff end is going to vibrate in response to high notes, whereas the wide, more flexible end will vibrate in response to lower notes. Now we know that sound travels in waves. High pitches travel in quick waves. Low pitches travel in these slow waves. And so this is how that basilar membrane is reacting differently to different pitches. It depends on the waves that are coming in. Now, depending on which end of that membrane is moving, that is the end that those hair cells are going to be stimulated on. So if the narrow stiff end is moving, then the hairs on that end will be stimulated. If low notes are coming in and the wide flexible end is moving, then the hair cells on that end will be stimulated. And so your brain is interpreting signals from different hair cells as a specific pitch. So now we know how we distinguish pitch. How do we distinguish intensity or loudness? Loudness depends on how much vibration is happening in a particular portion of that basilar membrane. The louder something is, the more vibration, more deflection of that basilar membrane, and that means more hair cells are stimulated. Now, really loud music, loud sirens, even, I know this sounds weird, but they've done some tests and they see that if you go to the movie theater and you watch a movie that has like an action movie where there's really loud noises in the movie, sometimes the intensity of that sound can be louder than a jet engine. So movie theaters are really, really loud sometimes. If you are constantly exposed to really loud noises and that basilar membrane is deflecting a lot and moving a lot and hitting and slamming down on those hair cells all the time, those hair cells can become damaged. And that can lead to partial deafness. Those hair cells don't regenerate. So this is why people who work outside with planes, for example, like air traffic control, they wear ear protection. People who cut lawns, they wear or should wear ear protection because that is really loud, and over time it can damage those hair cells and it can cause deafness. Now, in terms of hearing loss, we actually have different kinds of hearing loss. There's a type of deafness called conduction deafness. And this happens when you cannot conduct sound waves into the inner ear. This might be caused by a rupture to the eardrum. So when those sound waves come in and they normally bounce off the eardrum, if the eardrum's ruptured, if it has a hole, it's not going to vibrate very well. That means that what you hear isn't going to be as loud or as good. If you have a ruptured eardrum, things sound muffled. It almost sounds like you're underwater. Or damage to those ossicles. So I know we talked about ear infections, and repeat ear infections can cause those ossicles to fuse together. And if that happens, then they can't vibrate in response to sound waves coming in. That leads to conduction deafness. How conduction deafness is treated is through hearing aids. Hearing aids basically amplify the sound. They make it louder so that you can hear. The other kind of hearing loss we have is called nerve deafness. This is physical damage to the nerve or the hair cells. So, being exposed to really loud noises for a constant long period of time damages those hair cells. They do not grow back. That's nerve deafness. Damage to the vestibulocochlear nerve, or even damage to that portion of the brain, that cortex called the auditory cortex. That results in nerve deafness. There is no way to treat nerve deafness. Hearing aids do not help. You've damaged the nerve. It doesn't matter how loud we make the sound, you're still not going to hear it. So we've walked through hearing in the ear. Let's talk a little bit about balance, because remember, those semicircular canals are heavily involved in balance and body position. So the vestibular apparatus deep inside the brain is going to house the semicircular canals, the utricle, and the saccule. Now remember, your semicircular canals are filled with fluid, and when that fluid moves, it tells the brain that you are moving. This is going to help us stay balanced when we're moving. The utricle and the saccule, so remember these are in the vestibule, these also help us stay balanced. They help us stay balanced during rest and during acceleration. Now the utricle and saccule are actually pulling information from our eyes, our joints, and our skin to help with balance. So believe it or not, all of these help your body understand its position and whether or not it's moving. So a great example of this is if you've ever been out on a boat in the middle of the ocean and you've gotten seasick. Typically, if you go on a boat in the ocean, they tell you, don't look down at the ocean. Don't look down. Because if you're looking down, a lot of times your eyes are just looking at the water, and your eyes are not registering the amount of motion that you're actually experiencing on that boat going up and down in the swells. And so your eyes are telling your brain, we're not moving. But the vestibular apparatus, like those semicircular canals, they're telling the brain, oh yeah, we're moving a whole lot. And it gets pretty confusing, and it can result in nausea. And so a lot of times if you're out on a boat in the ocean, they'll tell you if you start feeling a little bit queasy, you need to look up and look at the horizon. Because if you look at the horizon, or if you look at land, then your eyes adjust and you can identify, oh yeah, visually you can tell your boat's going up and down in the swells, and your brain is getting the same information from the semicircular canals as it is from your eyes. Now this is a great picture. This is showing what the inside of the semicircular canals look like. And you can actually see all these little tiny hair cells in here, these little hair bundles. And this is all filled with fluid. So as you're spinning, like for example, during a rotational movement, you can see that that fluid is rotating and it's causing those little cells to bend. If it flows the other way, you can see they bend the other way. So that is it for Chapter 15 lecture. We focused on general senses that relate to touch. So this is anything from pressure and temperature and pain. And then we focused on our special senses, which are everything else. So we talked about sight, taste, smell, and hearing.